السلام عليكم ورحمة الله. Welcome شباب, welcome my dear sons, students of grade first secondary. We continue talking about the nucleic acid today, and we will solve some questions in this session. We'll start solving some questions about, inshallah. We will revise it quickly. We said before that nucleic acid is a type of biological macromolecules. It is a polymer which is made up of smaller units called monomers, and the monomers of the nucleic acid are called nucleotides. Nucleotides. Okay, so we have the polymer, which is nucleic acid, and the monomer, which is called a nucleotides, the monomer of the nucleic acid. And the most famous nucleic acid, which we studied before, DNA, and also we have RNA. The most famous nucleic acid, DNA and RNA. And they contain, because they are biological molecules, they contain, of course, carbon, hydrogen, and oxygen as basic elements. And also, there is nitrogen and phosphorus. Also, they are basic elements. So we have 5 basic elements in the structure of the nucleotide and in the structure of the nucleic acid. Let's see where is the nucleic acid found. We can see here the nucleic acid is found in the chromosome. We know that the nucleic acid is found in the chromosome. We know the genetic material, المادة الوراثية, the genetic material is found inside our body in the nucleus of the cell. And there is, in the nucleus, there is a chromatin or chromosomes. The chromosomes they carry the hereditary traits. How they carry the hereditary traits? They carry the genes which are responsible for appearance of the hereditary traits. And the genes are found on the DNA, on the DNA molecule. The DNA molecule is a very long thread, double thread. We say it is called double helix. And it is made of a long thread, it's very long thread, and it's made of many sections. And the gene is a part of DNA. You can say this is a gene, and this is a gene, and this is a gene, and so on. So DNA is a very long thread, very long molecule. You can divide it into parts. These parts are called genes, and every gene is responsible for appearance of a certain hereditary trait. But what is the structure of the DNA? The monomer of DNA, or the units which the DNA is made of, is called the nucleotide. The nucleotide is the building unit or the monomer of DNA. We need to know the structure of the nucleotide. It has 3 main basic structures. We can divide it into 3 main basic structures. As you see here, we have a nucleotide. You can magnify it like this. The nucleotide is made up of 3 components or 3 parts. The first part is a sugar, a molecule of sugar, pentose sugar. As you see here, this is the pentose sugar, the first part of the nucleotide. It may be deoxyribose in the case of DNA, and it may be ribose sugar in the case of RNA. The difference, there is lack of one oxygen in the case of deoxyribose, which is found in DNA. This is the first component of the nucleotide. The second component of the nucleotide, which is the phosphate group. It is joined to carbon atom number 5 here. The phosphate group is joined to carbon atom number 5 in the pentose sugar. And finally, the third component of DNA, which is the nitrogenous base. The nitrogenous base here we have connected to carbon atom number 1 of the pentose sugar. So we have 3 main components in the nucleotide. We have pentose sugar, سكر خماسي. We have phosphate group connected by a covalent bond to carbon atom number 5, and we have nitrogenous base connected to carbon atom number 1 also by a covalent bond. So we have 2 covalent bonds. We have covalent bond here between the sugar and the carbon atom, between carbon atom number 5 and the phosphate group, and we have covalent bond here between carbon atom number 1 and the nitrogenous base. Okay, let's talk about the differences of the function and the differences of a DNA. When we are making a DNA molecule, we make a long chain of nucleotides. When we are making a long molecule of DNA, we make a bond between nucleotides together. The nucleotides bond together by covalent bonds, where they bond together by covalent bond at the nitrogenous bases. The nitrogenous bases bond together on the molecule of DNA. Here we have nitrogenous base, for example. We have here nitrogenous base, and we have here another nitrogenous base. They bond together by what? They bond together by a covalent bond, maybe double covalent or maybe triple covalent. We will talk about it now, but we have to know that the nucleotides bond together by covalent bond, maybe double and maybe triple. We will discuss it after a few seconds. So we are going to structure of the DNA, talking about the structure. Here we have the structure of the nucleotide, three main components. Remember, we have three main components in the nucleotide. The first component, which is the pentose sugar, which is bond with a covalent bond with nitrogenous base. This is the second component, and bond with also a covalent bond with a phosphate group. The phosphate group bond with the pentose sugar at carbon atom number 5 by a covalent bond here. And the nitrogenous base bond with the pentose sugar by a covalent bond here in carbon atom number 1. So this is the structure of the nucleotide, the monomer of the DNA. Now, if we want to bend two nucleotides together, if we want to bend between two nucleotides together, what will we do? And here we have the differences between the nucleotide of DNA and the nucleotide of RNA. We have here nitrogenous bases, 4 nitrogenous bases, A, G, C, T. We have one, take letter A, which is adenine, and the one which has letter G, guanine, the one which has letter C, cytosine, and the one A with letter T, thymine. We have adenine, A, guanine, G, cytosine, C, and we have thymine, T. The 4 different nitrogenous bases. Okay, they are found. These 4 nitrogenous bases are found in each nucleotide in the DNA molecule. But in the case of RNA, there is no T. You can observe that we have here U and we don't have T. So the nitrogenous base uracil replaces the nitrogenous base thymine. Replaces the nitrogenous base thymine here. In the case of DNA, we don't have thymine, but we have a uracil. But adenine is found the same, guanine found the same, cytosine found the same. But the difference between DNA and RA in the 4th nitrogenous base, which is thymine in the case of DNA, and uracil in the case of RNA. Okay. Many differences between DNA and RNA. Look at this question. Which of the following is similar in DNA in presence of phosphate element, a phosphorus element in the structure? Which of the following is similar with the DNA in the presence of phosphorus element? Phospholipids, glycogen, casein, A and C. Of course, the answer will be A and C. A and C. Because phospholipid has phosphorus element, and the casein, the protein of milk, also has a phosphorus element. Let's move. We have here the compare between DNA and RNA. The nitrogenous bases First, DNA contain a pentose sugar called deoxyribose sugar. But RNA, the sugar is called what? The sugar is ribose sugar. The difference here, there is lack of one oxygen molecule. There is lack of one oxygen molecule or atom, oxygen atom in the ribose sugar. So we call it deoxy. Deoxy, this means there is one oxygen atom less, one oxygen atom less than the ribose, the normal ribose sugar. So we call it deoxyribose sugar. This is the word DNA comes from, deoxyribose sugar. But RNA come from ribose sugar, so we call it ribonucleic acid. But here in DNA we call it deoxyribonucleic acid. The symbol DNA refers to deoxyribonucleic acid, and the symbol RNA refers to ribonucleic acid. So the difference here between DNA and RNA, the type of sugar. The type of sugar in DNA, which is deoxyribose sugar, and the type of sugar in RNA, which is a ribose sugar. This is the first difference. The second difference, the nitrogenous bases. The nitrogenous bases found in DNA, adenine, thymine, always bind together by double bond. Guanine and cytosine always bend together by triple covalent bond. So adenine always bend to thymine with what? With double covalent bond. Guanine always bend with cytosine with what? With triple covalent bond. And we have here in the case of RNA, adenine bend with what? Bend with uracil, no thymine. So adenine bend with uracil, another nitrogenous base, replaces the thymine, and it bend with adenine by double covalent bond. And we have also guanine bend with cytosine with triple covalent bond. This is the second difference, the type of nitrogenous bases. So the difference here, it is clear. We have here thymine and we have here a uracil. This is a big difference, thymine and a uracil. Thymine in DNA and uracil in what? In RNA. Let's move to another point. Number of strands. How many strands of DNA? The strands of DNA, two strands, double strands. There are two strands or double strands in the case of DNA. But in RNA, we have one single strand. We have single strand, one strand only. You can look at the picture of DNA and RNA here. We have double strands, double helix, or double strands. It is double thread. And here we have single thread only. Single thread only. So we have here the difference between DNA and RNA. And remember also, DNA is found only in the nucleus of the cell. But the RNA can found in the nucleus, and it can move outside the nucleus, go to the cytoplasm, carrying the genetic information, carrying the information which we can use to make protein or to make enzyme or to make something like this, hereditary trait. So DNA found only inside the nucleus, but RNA can found in the nucleus and can transfer outside the nucleus of the cell. Also, the function of DNA, it carries the hereditary traits. It carries the chromosomes inside the nucleus of the cell. Whereas it carries the genetic information. It carries the genetic information that passes from one generation to another. When the cell divides, the information passes with the chromosomes from parents to offspring. So it is responsible for appearance of the hereditary traits. All characters and all traits appear due to the genes. Genes are found on DNA. So it is responsible for appearance of the hereditary characters of the living organism, and it's responsible for organizing all vital activities of the cell, carrying hereditary traits and organizing all activities. But the function of RNA is different. It transfers from it transferred from DNA. You can say that RNA is a copy of DNA. It is only a copy of DNA. Part of DNA is copied and you make RNA. It is transcribed from nucleic acid DNA inside the nucleus. Then it transfers into the cytoplasm to be used in building or synthesizing the protein in the cell. The protein is used in making maybe a hereditary trait, or making an enzyme, or making the hormone, or something like this. So it is responsible for appearing of the genetic traits, and also organizing all vital activities. So you can say that RNA is only a copy of a part of DNA. It is transcribed from a DNA. Now we can go to the exercise about the nucleic acid. We want to solve some questions about the nucleic acid. We have here multiple choice question. A part to represent the carbohydrates is the structure of RNA. Is it uracil, or ribose, or adenine, or deoxyribose? We are talking about RNA, so we are talking about ribose. We are talking about ribose. The ribose sugar represents the carbohydrates in the structure of RNA. Now the number two, the number of hydrogen atoms in the sugar that enters the structure of DNA. Again, the number of hydrogen atoms in the sugar that enters the structure of DNA. We remember that DNA is a pentose sugar. The chemical formula of DNA, we know that it is C6H12O6 in DNA. But this is the glucose, but the ribose, it will be C5, not C6. Remember, the glucose is C6, but here we have 5 carbon atoms in the pentose, because it is pentagonal or pentose sugar, سكر خماسي. So the number of carbon atoms will be what? If carbon atoms is 5, so the number of hydrogen atoms will be what? You remember the ratio 1 to 2 to 1. So hydrogen will be double of it. It will be 10. Hydrogen will be what? Hydrogen will be 10. The answer will be 10 hydrogen atoms. Okay. If carbon was 5, so the hydrogen will be 10. Okay. Number 3, the molecular structure of ribose sugar. The molecular structure of ribose sugar. Of course, 5 carbon atoms and 10 hydrogen atoms. 5 carbon, 10 hydrogen, and 5 oxygen. So the answer will be what? The answer will be A. The answer will be what? The answer will be A. Number 4. Now we can go to number 4. By using the following figure, which of the choices that are represented in the table is correct? You can see this. We have a polymer here. We have here a polymer. Maybe this polymer is DNA, okay, or RNA. This is made of monomers. Well, we studied many polymers. Carbohydrates are polymer, lipids are polymer, proteins are polymer, nucleic acid are polymer. So we have polymer made of monomers. So here we have a polymer in the left side and in 1, and we have monomers in the right side in 2. So let's choose which of is correct. Amino acid and polypeptide chain. Amino acid, the protein monomer, and polypeptide chain is the polymer. The left side is not a monomer. It is a polymer. Okay, let's see. So number 1 is wrong, because he says here a monomer and here a polymer, and we have here 1 is a polymer and 2 is a monomer. Okay, let's see number 2, B. Polypeptide chain and fatty acid. This is a polymer and this is a monomer. But is the polypeptide chain made of monomer fatty acids? Of course not. The polypeptide chain is made of monomers, which is a amino acid. So number 2 also, number B is wrong. Okay. Now let's move to number C. nucleotides and DNA. Nucleotides are monomer, and we have here a polymer. And here DNA is a polymer, and we have here monomer. So number C is wrong also. And now finally we can find number D is correct because DNA is a polymer, and here we have a polymer. And number 2 is nucleotides, which are the monomer. This is easy. So the correct answer is A, D. Let's move to another question. Nucleotides differ from each other. Nucleotides differ from each other in the following, except building of structure of glucose. Nucleotides differ in the building of the structure of glucose. All nucleotides have fructose sugar. So number 1, this is wrong. Okay, this will be wrong. Okay. The type of nitrogenous base, of course, every nucleotide differ from each other in nitrogenous base. So we will not choose the type of nitrogenous base. The phosphate group, every nucleotide has phosphate group. So the correct answer will be what? The site. It is the site, the difference in the site. There is no difference in the site. Okay. The answer will be site. This is the correct answer. Let's move to number 6. The number of nucleotides that are common between DNA and RNA. The number of nucleotides common between DNA and RNA. You remember that DNA have nitrogenous bases A, T, C, G. Okay, this is the nitrogenous basis of each nucleotide in DNA. And RNA we have A, U, C, G. So how many nitrogenous bases are common? How many similar nitrogenous bases? So will be similar in the DNA and the RNA. We have only one here, not found here. So if we remove T and U, we have 3 similar nitrogenous bases. So the answer will be 3 common between DNA and RNA. 3 nitrogenous bases and 3 nucleotides are similar or common. Okay, the answer will be 3. Let's move to another question. If you know that the adenine A bind with thymine T and the guanine G bind with cytosine C in a double helix of DNA, in the light of this, enter the following. The percentage of is equal between each of the following. The percentage is equal between each of the following. Adenine and guanine, of course not. Adenine doesn't bend with guanine. Thymine and cytosine, cytosine doesn't bend with cytosine. Adenine and thymine, yes, here this is correct. Adenine and cytosine always bend together, so they have the same number. They have the same percentage. Adenine and thymine. But A and B is wrong. So we have here the correct answer will be adenine and cytosine. After that, we have which of the following graphs doesn't express the DNA structure according to the nitrogenous bases. Okay, doesn't express the DNA structure according to nitrogenous bases. Okay, we have to know that DNA has a similar number of A and T. Similar number of A and T. Okay, similar number of C and G. A and T similar number, and C and G similar number. So we have here similar number of A and T and similar number of C and G. Okay, this one may be correct. This one may be correct. Here, but let's focus to the other one. A and T and C and G. We have different numbers, but A equal T and C equal G. The number, the percentage of A and T equal, and the percentage of C and G equal. Let's move to number C. A, C, T, G are equal percentage. I think this is the correct number. Here we have the correct answer. We have the answer will be C, because we have equal percentage of A and T, C and G. This is the correct answer. This is the correct answer. Let's move. The opposite diagram expresses a part of the nucleic acid DNA. What do the numbers 1, 2, 3 represent respectively? If you remember the structure of the nucleotide, you can see we have three main components. A pentose sugar like this. Here number 2, we have pentose sugar, and number 1, we have nitrogenous base, and here we have phosphate group. So the arrangement, the correct arrangement, 1 is what? 1 is nitrogenous base. 2 is what? 2 is a pentose sugar. 3 is what? 3 is a phosphate group. So let's see which is correct. Nitrogenous base, phosphate, pentose sugar, and phosphate group. So the correct answer will be C. The correct answer will be C. Let's move to number 9. The nitrogenous base T is similar to nitrogenous base U in the A in space. They have the same site or the same structure or the same shape or the same sugar to which they bend. T and U similar in what? Similar in what? Yes, they are similar in size. Yeah, they are similar in size. This is correct. This is correct. They have the similar size. They have similar size. The different T found in sorry. They are similar in T found in T found in DNA and U found in RNA. D found in thymine is found in DNA, but uracil found in RNA. So different in site. They have both sides the same site, but they are different in what? They are similar in what? What is the structure? Similar in what? They similar in they are not similar in the sugar, of course, because T bend to deoxyribose sugar and U bend to ribose sugar in DNA ribose sugar. So we can remove the incorrect answers. The site is wrong, the sugar is wrong. Now we have structure and shape. Are they different in structure or similar? They different in structure, of course. So they can be similar only in shape. They can be similar only in shape. The correct answer will be shape. Okay. Okay. The last question: nucleotide containing the adenine in DNA. The nucleotide containing adenine in DNA molecule differs from nucleotide containing adenine in RNA molecule in the number of what? The nucleotide of DNA, you know that the nucleotide bend to deoxyribose sugar, and the nucleotide of RNA bend to ribose sugar. And the difference, you remember, the difference between deoxyribose and ribose, the difference in the number of the oxygen. Very good. they have one less oxygen atom. There is a lack of one oxygen atom. The correct answer will be oxygen. Okay. I will end the session now, and I let you continue solving the other questions, and send me your answers. Thank you, everybody. Until we meet again. Assalamu alaikum wa rahmatullahi wa barakatuh.